So our first speaker is Seungmae Seo, who's going to be talking to us about an iPS-based model of natural killer cells and a very sort of interesting situation between related individuals with very diverse phenotypes. So May, go ahead and take the screen share and go for it.
Hello, my name is Seungmae Seo. I'm an associate research scientist with Dr. Emily Mace at Columbia University. As Ron has mentioned, I had worked with Ron, Carlo, and Suki on the variation in pluripotent stem cells that was presented last month. And then I decided to continue my work in hematopoietic system, and that's the work that I'm going to present today. I'll be talking about how we are modeling hematopoietic monogenic disease using iPSCs. And for this seminar series, I want to specifically talk about an aspect of this project where we do not have all the answers yet, but we observe human variation interacting with disease-causing mutations that lead to different phenotypes. I want to end the talk by discussing how these observations may guide our future studies. As I understand, this is a very neuroscience-heavy audience, so I'm going to do my best to start slow and have some more introduction. Let's start with our main character. So our lab is interested in understanding the development and functionality of natural killer cells. As the name suggests, they are responsible for killing infected or diseased cells without prior sensitization to them. When the NK cell encounter a target cell, the receptors will start interacting with the surface proteins of the target cells. Once the NK cells recognize the target cell to be foreign or virally infected, it'll get activated and create immune synapse. Immune synapse can be seen in this microscope image on the right-hand corner, where NK cell is on the top and target cell is at the bottom. The target cell is then killed by a process called degranulation. It's very similar to the process to how neurotransmitters are released. During degranulation, instead of neurotransmitters, and proteins like perforin and granzyme, which can break down the target cell's membrane, and proteins are released. So we use this expression of these proteins, perforin and granzyme, as markers of NK cell maturation and functionality. There are a few ways that NK cells are made, especially during development, but the most well-characterized pathway is in the adult hematopoiesis. In adults, NK cells originate from the bone marrow hematopoietic stem cells. During their development, the NK cell progenitors move to the correct environment to differentiate. And as represented in this diagram, the early NK progenitors migrate to secondary lymphoid tissues such as tonsil, lymph node, and spleen. And they re-enter the bloodstream after maturation as a mature NK cells. You probably know that the T cells do a similar thing where they move out of the bone marrow and mature in thymus. And it's a very similar process. NK cells are continuously regenerated throughout life, and this process has been described in stages according to their expression of surface markers as listed here. The markers highlighted in red are the expressed ones for each stage. And you can see that there's a sequential acquisition and loss of these surface markers. We'll be using many of these markers in our experiments to track NK cell differentiation from the iPSCs, and roughly we'll refer to the intermediate NK cell progenitors as stage 2 and 3, and more mature NK cells, similar to the ones that are in blood, to be stages 4 and 5. So in flow cytometry of peripheral blood mononuclear cells, NK cells are identified as CD3 negative, CD56 positive, and they comprise 10 to 20% of the cells. NKD stands for natural killer cell deficiency, so obviously that means that the individuals are missing those NK cells. Most NKD presents with a lack of NK cells, as you can see here, but the other populations in the blood are untouched, as you can see, outside of this box. Often the patients are admitted to the hospital with severe recurrent viral infections. Now, how frequent do we observe NKD? NKD is very, very rare. There are about 500 reported genes that cause primary immune deficiencies, where specific immune subsets are missing. About 10% of them affect NK cells as well as the other immune compartments. There are 8 genes that are known to cause NKD, where NK cells are specifically impaired, and we're going to look at one of these cases. While it's extremely rare, 4 of the 8 genes highlighted in red here belong to the same DNA helicase complex, indicating that there could be an underlying NK cell-specific biology related to helicase activity. So as you all know, helicase is a protein complex required for eukaryotic DNA replication. During G1 and S phase of the cell cycle, helicase proteins assemble at the origin of the replication, as you can see in this diagram. Here are the 4 reports that talks about the 4 genes causing NKD and 4 mutations causing NKDs. And there are common features of these helicase variant-induced NKD phenotypes. They first show decreased NK cells in the periphery, and they show specifically decreased frequency of the most advanced, most mature stage 5 NK cells relative to stage 4 NK cells in the peripheral blood. They show impaired NK cell functions and little to no involvement of T cells and B cells. This last point is important because T cells and B cells share common progenitor with NK cells. So what this means is that the helicase variant cause defect that occurs later in development after the cells have committed to NK cell lineage. Here is a summary of our patient phenotypes. So on the left side is our proband. Our proband was admitted to the hospital at 11 months of age with generalized seizure. And he suffered all these listed infections during the first 3 years of his life. His blood was tested and it showed severe neutropenia and NKD. He received GCSF treatment, which helped him with neutropenia and managing the infections, but NKD is still not resolved. His younger sister was tested early for immunodeficiency, and she was found to have moderate neutropenia and NKD. She also went on GCSF treatment for 2 years, which stabilized her neutrophil count, but NKD is still there. Without reading the details, you can immediately see the different severity of the disease between the proband and the sister. And we'll come back to this and talk about how this shows up in our culture system later. Here are the flow cytometry plots of their PBMCs, all the family members and unrelated controls. The red numbers and boxes represent NK cells. Our proband and the sister, as you can see, show significantly reduced NK cells compared to both of their parents and unrelated donors. From exome sequencing, all 4 of the family members were tested, and we identified variants on a gene called GENS4. Both father and mother had point mutations, and especially mother's mutation led to a premature stop codon. The proband and sister were compound heterozygous who inherited both of these variants. As a result of these variations, they present with about 15% of GENS4 protein in their cells compared to the control. To study the pathogenesis, we generated iPSCs from all 4 of the family members. As you can see here in the Western blot, the iPSC showed lower GENS4 level, just like the primary cells. We also used CRISPR to correct both of the GENS4 variants in the proband line, and upon correction, GENS4 protein level was rescued. Since GENS4 is a cell cycle-related gene, we looked at cell cycle profile using EDU and DAPI, but we did not observe any cell cycle defects in pluripotency. For differentiation, we first made CD34 positive hematopoietic progenitors in embryoid bodies. As you can see here, it takes about 6 days to get to this point. And then for in vitro natural killer cell differentiation, the progenitor cells require 2 things. One is the stromal cells for support, and second is a series of cytokines that's been listed here. We carried out our experiment in 2 conditions with different stromal cells. First was with OP9. These are the cells derived from the mouse bone marrow stromal cells. And this is widely used stromal cell line for in vitro NK cell or any other hematopoietic differentiation. from the hematopoietic stem cells. Second is an autologous stroma cells. So this is an iPS-specific stroma that can be generated. These are the cells that grow from the non-CD34 positive cells within the embryoid body. So we are basically plating the entire embryoid body on a plate and let it grow, and CD34s will become hematopoietic lineage, and CD34 negative will become the stromal cells. We collected samples every week and characterized all the intermediate cell types that lead to the NK cell generation. So here's what differentiation of a control line on OP9 looks like. On day 6, we sort CD34 positive hematopoietic precursors. Day 21, majority of the cells express CD45, showing hematopoietic commitment. On day 28, we start to see hints of CD56 positive NK cells appearing. And on days 34, 35, and 42, proportion of the NK cells increase, and these NK cells start expressing more markers indicating further maturation. The flow cytometry data, I'm showing six antibodies right now, but the flow cytometry data contains more than those markers. So I'm summarizing the data into a TSNE here. This contains cells from all time points and all cell lines. And as you can see, we have they can be clustered into 6 different groups. We have pink and gray, which are non-NK hematopoietic cells, and then 4 NK clusters of varying maturity. Heat maps here show that the expression of marker genes, especially the maturation markers, which is on the right-hand side, you can see that these markers are being expressed in the more mature NK cell clusters as they appear in the later time points, peaking at day 35. So then obvious next question is what happens when you differentiate genes for variant lines? First, here is the same TSNE plot that contains all the cell lines and all time points. If we just look at the control lines and the parent lines, you see non-NK clusters appearing first. That's the pink and the gray clusters over here. And then NK clusters show up between days 28 and 35. And the parents and the unrelated controls are comparable in these time points. With genes for variant lines, however, this is what we see. First, you would notice that there is a significant decrease in cell survival. And then you see that there's an absence of NK cells at all stages of development. So here we confirm that iPSCs recapitulate patient phenotype by not generating natural killer cells. So then what happens to the autologous stroma? In grand scheme of things, it's very similar to what happens to OP9. On day 6, we have CD34 positive precursors. On day 14, we start to see CD45 suggesting hematopoietic commitment. On day 21, we start to see the hint of CD56 positive NK cells. And later on day 28, we see the majority of CD45 positive cells are NK cells, expressing intermittent maturity markers, CD117. And later on days 35 and 42, we see maturation of NK cells to express more CD94 and granzyme B, the cytotoxic protein. Similarly, again, TSNE plot here shows 9 clusters now. Now, this one includes 2 of the autologous stromal cells, that's the blue and red. These are the CD34 and CD45 negative cells. So therefore, they are not non-hematopoietic cells that's in the culture. And then we have CD34 positive cells in the brown cluster. That's the day 6 cells. And then we have the purple, pink, gray over here. These are the non-NK hematopoietic cells. And finally, we have NK cells in orange and green. Oops. I don't think I can. Sorry, let me try to mute. Okay. So now looking at individual lines on the autologous stroma differentiation, if you just look at the control line across time, similar to the OP9 differentiation, we see emergence of the non-NK cell NK hematopoietic clusters first, and then NK cell clusters showing up between days 28 and 42. We collected the CD45 positive cells from day 42 and confirm their functionality as NK cells by cytotoxicity assay. With genes 4 proband line, however, this is what we see. Up to day 21, everything is very comparable to the control, but between day 28 and 42, instead of the NK cell emerging, we see the stromal cells persisting. And of course, if we collect the CD45 positive cells from this day 42, it's these tiny amount of cells, we see no killing of the target cells since they are not making NK cells. With the CRISPR correction line, we observe restoration of the NK cell emergence as well as the killing in cytotoxicity assay. So here we're confirming two things. One is that again, iPSCs recapitulate patient phenotype by not generating NK cells. Number two is that we observe the isogenic control rescues the NK cell potential, suggesting that genes 4 variants are causing the NKD phenotype that we have been observing. I'm skipping a lot of the other experiments, but here to show you the most important finding, we surveyed cell cycles throughout differentiation with EDU and Ki67. We identified NK cell-specific burst of proliferation, specifically on day 28. So we should be looking at the red histograms here. Those are the NK progenitors. As the red arrows show that on day 28, these NK cells, as they start to emerge, they had burst of proliferation. Repeated studies show that this burst was missing in the proband line and came back with the CRISPR correction, suggesting that there's a specific time point and specific proliferative burst that's affected by the lack of genes 4. So our current model is that when early NK cells emerge, the proband cells with lower genes 4 levels cannot proliferate, and that ultimately leads to the limited or no generation of NK cells in the end. Now, this is the most interesting portion of today's discussion. Having the same variants, the proband and the sisters are not exactly the same in our experiments. In these bar graphs, I'm showing the efficiency of NK cell differentiation. Each bar represents individual experiments, and the darker colors represent more differentiated NK cell identities. In the bottom, the horizontal bar represents cell lines. So you can see that in this control line, you can start to see the line-to-line and cell and experiment-to-experiment variations. In this control line, you can see 5 out of 6 times the cells differentiated to NK cells quite efficiently on OP9. However, here are the proband and the sister, and you can see the dramatic decrease of differentiation efficiency that they are experiencing on OP9, which was rescued with the CRISPR correction line. On autologous stroma, something interesting happens. The proband is inefficient like OP9, like they were in OP9 culture, but the sister line is doing just fine generating NK cells. The NK cell generated here are transcriptionally indistinguishable compared to those from the control lines, and their functionality is also comparable in killing assays. Moreover, our proband lines sometimes do generate NK cells efficiently. And when we look at the cells generated in these rare cases, they look just like mature NK cells generated from the control lines, expressing these maturation markers such as CD94 and granzyme B. So what does that mean? I want to remind you that the NKDs present with decreased NK cells, but not zero. And the sister consistently have more NK cells than the proband. And she also has less severe overall clinical phenotype. So together, what these data seems to suggest is that the lack of genes 4 is creating a bottleneck in NK development without affecting the potential itself. And there appears to be some epigenetic state that can overcome this bottleneck, which seems to be more prominent in the sister. Currently, we do not understand how the bottleneck is controlled and how the sister line can bypass that, specifically in the autologous stroma condition. But however, we should be able to approach that mechanisms by experiments where we mix the cell lines or perhaps looking further into transcriptional signatures to understand where the differences exist. Identifying the mechanisms of overcoming this genetic bottleneck will give us hints on how to develop potential treatment that may be generated to all helicase, may be generalized to all helicase-induced NKDs. We're looking forward to expanding this work using additional patient-derived iPSC lines and more control lines. As we have been hearing throughout this seminar series, looking at multiple cell lines and trying to understand their distinct behaviors in DISH will help us gain a broader understanding of variation in human development. And on top of that, introducing genetic diseases like NKD would reveal how different genomes or epigenomes interact with mutations to result in pathogenic variabilities as well. And with that, I'd like to thank my colleagues at the MACE Lab and Orange Lab, collaborators at Columbia University and Baylor College of Medicine, and our funding. I skipped a lot of experiments, but the preprint is available on bioRxiv. And if you have any questions or comments, please feel free to email me at any time. Thank you very much.
Thanks, May. And any questions?
Well, Carlo, if nobody else has a question, maybe I can just ask you a question, May, that I've had the pleasure of listening to earlier versions of this. But what I find myself focusing on here is this autologous stroma experiment. So you present it as if it's just some sort of random cell, but actually, isn't it a sort of hematoendothelial cell? It's a different cell in the early lineage. Or am I not getting this?
So the autologous stromal cells are not endothelial cells. However, they do show mesodermal lineage, and they show specifically cardiovascular signature quite strongly. They express GATA4, 5, 6 at different time points. And as we differentiate the cells, the autologous stroma also seems to change. And we have RNA sequencing data showing that they express their transcriptional signature change across time, and they move around between mesenchyme and extraembryonic endoderm and cardiovascular cells. So it's very interesting to think about what are these cells and how are they supporting.
Okay, what I was saying is they appear to complement the deficiency.
Right.
Right. That's what's so stunning about them. They're actually very interesting cells, these cells, from your mechanistic point of view.
Yeah. Yeah.
I think that's true in the human. It's the same mechanism that you're seeing in the dish and that is occurring in this in the sibling.
We don't know. Right. So it'll be very interesting. It would be very interesting to see, like, if it's really true. Like, does her bone marrow have different microenvironment, and that kind of thing would be very interesting to see. And we do not know what exactly is happening in the in vivo system.
May we also have a question in the chat from Victor Luria.
Okay.
What does the truncation in GINS4 do? Which domain is affected?
So the mutations are at the end of the protein. So mutations at the C-terminal end of the protein, and the father's mutation is just a point mutation, valine to leucine. I'm not sure what it does, but the father's phenotypically is fine. The mother's mutation is a early end codon generation at the end of the protein. So what we think, and she has about 50% of the protein compared to the control. So what we think is that her mutation induces degradation of the proteins, like nonsense mediated decay kind of way. Thank you very much.
Thanks, May.